An analysis of the swine flu genome sequence by Alexander S. Jones, an NIH whistleblower, indicates that only 5% of the novel swine pandemic influenza A RNA sequences, swine flu combined with avian flu virus identified in the pandemic 2009 virus, share no known homology in any public databases, in addition to the avian-swine hybrid nature of both these critical genes, and so a laboratory origin for this virus is the only logical conclusion. 95% if the sequences are readily available to microbiologists working in departments of defense weapons labs. Example, Fort Detrick and can be spliced together to form a nearly perfect match to a previously identified viral pathogen. It is public knowledge that Dr. Jeffrey Taubenberger's team at Fort Detrick painstakingly identified each genetic code on the 8 RNA gene strands of the 1918 killer virus. Once identified, the next step in reverse engineering the 1918 killer virus was to access the U.S. Institute of Pathology's massive bank of live virus samples to begin the splicing of the RNA strands, and placing them in plasmid cells. Taubenberger's task was completed in 2003, and the deadly 1918 virus was successfully cloned and added to the biological weapon arsenal at Fort Detrick, Maryland. These are supposedly the computer-generated closest RNA matches of the gene clades identified by Taubenberger's team in 1918 virus, with the existing gene sequencing on computer files at the World Health Organization databanks being a perfect match. These existing flu gene sequences were in fact systematically reasserted and inserted into plasmid cells to form the current pandemic virus by Taubenberger and his team. It is the sworn testimony of 3 independent virologists that this novel gene clade sequencing could not possibly have happened spontaneously and naturally. There are 36 hemagglutinin spliced protein sequence homologies identified in the novel 2009 swine pandemic flu, as well as 30 NS1 non-structural protein sequences, for a grand total of 66 unique gene structures that have been reasserted and spliced together to form this reverse-engineered pandemic virus. 5 hemagglutinin and 1 neuraminidase molecules, a total of 6 molecules, were thus reverse-engineered by means of the 66 spliced protein sequences. In CIA and Mossad intelligence circles, the bioweapon was codenamed 666 bird flu, 6 molecules altered by means of 66 spliced RNA proteins. Alexander S. Jones concluded, "We must seriously consider a laboratory origin for this virus, because 5% of both these influenza A RNA sequences share no known homology in any public databases. If it was truly a novel antigenic shift, the vast majority would have no known homology gene sequence in viral databases. Blast sequence homology of swine flu indicates both the hemagglutinin surface protein as well as the non-structural NS1 interferon. Inhibition proteins are novel recombinants, previously unidentified in nature. Both these influenza proteins, H and N, based on the genetic sequences released Friday, May 1st by the U.S. Centers of Disease Control, share their closest genetic identity with turkey, avian, and pig, swine, strains from multiple continents, including North America as well as Asia. Even the closest matches indicate only 5% previously unidentified genetic material. I submit this evidence, coupled with the lack of presence of this virus at the pig farm near the proposed CDC's patient 0, a 5-year-old from La Gloria, 80 kilometers away from the pig farm in Perote, Mexico, shows that the origin of the flu outbreak remains unidentified at this time and cannot be ascribed to Mexican or North American swine in confined animal feeding operations. Furthermore, I submit once again that since 5% of both these influenza A RNA sequences share no known homology in any public database, in addition to the avian-swine hybrid nature of both these critical genes, that we must conclude that the virus originated in a bioweapons laboratory equipped with the tools to successfully gene-splice viruses. The bioweapons laboratory at Fort Detrick is the most logical suspect location of its creation. Future research that may be promising includes identifying critical SNPs, especially in the PB2 and the NS1 coding regions, which may be markers for evolution of pathogen virulence and should be closely monitored. The hemagglutinin protein should also be monitored for acquisition of a polybasic amino acid site, which would give the virus pantrophic properties as in the 1918 pandemic. Dr. A. Truott, PhD, ND, certifies to the best of his abilities that this virus is synthetic, laboratory created, and is not natural.
Streets are cleared away one by one. Hollow heroes separate as they run. You're so cold, keep your hand in mine. Wise men wonder while strong men die. Show me how it ends, it's alright. Show me how defenseless you really are, satisfied and empty inside. That's alright, let's give this another try. If you find your family, don't you cry. In this land of make-believe, dead and dry. You're so cold, but you feel alive. Lay your hand on me one last time. Show me how it ends, it's alright. Show me how defenseless you it's